Hello everyone and welcome back. A little bit of a heads up, this is a kind of involved chapter, and when I say involved I mean there's a lot of content to cover, as you'll see from the learning objectives that I have posted here, but there are several different things that we're going to be discussing. While you're taking notes for this chapter, some things that I want you to pay special attention to are, as I talk about each of these different classes of gram-positive organisms, I want you to specifically pay attention to their structure and physiology, their pathogenicity, the epidemiology of the disease, and then the few highlighted diseases, and also diagnosis, treatment, and prevention. So there are several different groups of organisms that we'll be discussing. So just as a bit of a heads up, you're probably going to want to stop this recording several times through in order to make sure that you're getting everything. In addition to that, I believe the recording, the PowerPoint slides, are about 102 PowerPoint slides in here, so we're going to do this in segments. This probably is going to be a 3- or 4-part lecture series. So I will be posting those up as we go through them. So without further ado, let's go here and look at some of these objectives that we'll be talking about. All right, so first and foremost, this chapter, chapter 19, is pathogenic gram-positive bacteria. So these are going to be all bacteria that are all gram-positive. So that means they're going to gram stain what color? Got that right, purple. They're going to be staining purple. There are two major groups of these gram-positive bacterial pathogens, and they are low G+C bacteria and high G+C bacteria. The names indicate that these classifications are based on the content of guanine, cytosine, nucleotide base pairs versus low adenine, thymine nucleotide base pairs in these organisms' DNA. Low G+C gram-positive bacteria have 3 genera of pathogenic spirococci: Staphylococcus, Streptococcus, and Enterococcus. There are 3 genera of pathogenic bacilli that are these low G+C gram-positive: Bacillus, Clostridium, and Listeria. The mycoplasma is a group that kind of sits on the outside of this because they don't have cell walls. So first we're going to talk about is Staphylococcus. Staphylococcus is a personal lab favorite because we use it a lot. It is part of your normal flora, so every member of the human race has some Staphylococcus on there. However, Staphylococcus can be what we consider an opportunistic pathogen, which means that they're able to, if the conditions are just right, they're able to cause disease. Now, one reason why Staphylococci is a good bacteria for your normal flora is that it can tolerate really high salt content. So for Staphylococcus, the structure and physiology, so I'm going to blow out my little sunburst here because this is some things that I believe are really important here. They are what we consider facultative anaerobes. And if you remember from previous chapters that we've talked about, something that's considered a facultative anaerobe means that it can use oxygen as the most efficient way to gain energy, but at the same time, it can also, if oxygen is not available, then it can also do without it. So it can also get energy through fermentation or anaerobic metabolism on here. Staphylococcus, we already know that the shape of them is going to be cocci because it says it right there in the name, it's Staphylococcus. So we see it right there in the name, it's cocci. And then staphylo is actually going to mean the arrangement. So we have these cocci, or these spheres, and the arrangement of them is that they're going to be in these grape-like clusters. They don't have a flagella, so they're going to be non-motile. As I said before, Staphylococcus are great organisms for the normal microbiota because they are salt tolerant, so they can tolerate the saltiness of the human skin. If you don't believe you're salty, lick yourself, or if you have licked someone else, what do they taste like? They're pretty salty on there. The saltiness of your skin, or that high NaCl content of your skin, helps it to retard most microbial growth, except for those that are in this Staphylococcus group, because they can tolerate that. Staphylococci are tolerant of desiccation, radiation, and heat, and they are even able to survive on various different types of environmental surfaces, such as your lab bench, if you do not properly sterilize it. So more structure and physiology. There are two commonly, I'm trying trying to move my thing around here. There are two species that are commonly associated with humans, and that would be Staph aureus, I'm trying to highlight this sucker for you, and Staph epididymis. Both Staph aureus, Staphylococcus aureus, and Staph epididymis, these are both organisms that we have worked with in lab. For Staph aureus, it's more of a virulent strain than Staph epididymis, and they can cause a variety of skin infections depending on where the site of the infection is. Staph aureus is also the base organism that causes MRSA, the methicillin-resistant Staph aureus. So there can be a host of infections that are caused by it. Staphylococcus epididymis is part of that normal microbiota that's on your skin, and this is the one that's more likely to cause infections for humans and be pathogenic when the opportunity arises. So if the host or the human's immune system is compromised, or if it's able to make its way from the surface of the skin into, and it's kind of says surface in its name, epidermitis, from the surface of the skin into deeper tissues, and if the immune system is compromised, as I said before, they can definitely cause an infection. So the pathogenicity of Staphylococcus aureus, that these infections typically take place when the organism that's usually meant to be on the outside of the body, like on the skin, it actually breaches the body's physical barriers and is able to get into underlying tissues. It only takes a couple hundred bacteria to result in a disease from it getting into the body itself. The pathogenicity of Staph aureus results from at least 3 different features of the bacteria. These are structures that enable it to evade phagocytosis. So it has these M proteins that allow for that to happen. The production of different enzymes, and also the production of toxins that will keep it from being properly dealt with in your immune system. So the structural defense against phagocytosis, they're coated with a protein that interferes with the antibody's immune response by binding to the different stems of the antibodies. These antibodies are meant to be as opsins, which means they coat the bacteria to make it sticky, and they're meant to enhance phagocytosis. But because of this binding, this outside protein that's on there, it keeps those antibodies from activating complement and causing opsonization to take place. There's also the outer surface of many strains of Staph aureus have this bound coagulase, and that enzyme converts blood protein fibrinogen into fibrin molecules, and then it will form blood clots around the bacteria. And then that will allow the blood clots that are formed around the bacteria allow for Staphylococcus to actually hide in those fibrin threads. So it actually takes your own body's defense system and causes clotting to happen, causes coagulase to happen, and it keeps the bacteria from being visible to the immune system. So there's structural defenses against phagocytosis. We talked about protein A, bound coagulase, which converts fibrinogen into fibrin and allows the bacteria to hide in there. And then the one that we hadn't talked about for a structural defense against phagocytosis are the synthesizing of these slime layers, or the capsules around it, and that prevents your white blood cells, or the leukocytes, from undergoing chemotaxis or being drawn towards the bacteria because they're kind of hiding in that slime layer. And then it also prevents the attachment of phagocytotic white blood cells like neutrophils or macrophages to eat that bacteria because of this slime layer. It's kind of slippery as a result. And the slime layer can also help for it to form really efficient biofilms because the Staphylococcus can actually attach to different surfaces. And we all know that a bacteria in a biofilm is much stronger in that community than it is by itself. So it will make your body's immune system have to work a little bit harder. So when we talk about pathogenicity, these are all ways in which the bacteria prevents itself from being detected or from being attacked by your body's immune system. And if it can't be attacked, if it can't be detected, then it will cause a disease. So enzymes that help with pathogenicity, there are several different enzymes that contribute to their survival rate. We have cell-free coagulase, and that's going to trigger blood clotting on here. Cell-free coagulase does not act on fibrogen directly, but it combines with the blood protein before that enzymatic conversion takes place. Only Staph aureus synthesizes this coagulase. Staph epidermidis and other species of Staphylococci are coagulase negative. So that's really important to remember that only, and I'll repeat again, only Staph aureus can synthesize coagulase. Hyaluronase breaks down hyaluronic acid. If you've taken A&P before, you're probably familiar with hyaluronic acid being the intercellular cement that kind of binds the cells together. By breaking down that hyaluronic acid or having an enzyme in there, it prevents the cells from having that nice bond, tight bond with them, and it can disrupt it and allows bacteria in between the cells, so that's going to further spread the disease or spread the bacteria throughout the body. Staphylococci are going to dissolve fibrin threads and blood clots, which will allow Staph aureus to free itself from the clot. So once it has escaped phagocytosis in the immune system, then it digests its way out of the clot. So once it escapes phagocytosis and it no longer needs to be hidden in that blood clot there, it can escape from out of it by breaking down the blood clot. Then we have lipases and beta-lactamases, beta-lactamases are present in about 90% of Staph aureus strains, and they can actually break down penicillin. Though beta-lactamases don't really have a role in inhibiting the natural defense of the body, it allows for the bacteria to survive when we use beta-lactam antimicrobial drugs like penicillin or cephalosporin. So lipases digest lipids, so Staphylococcus can grow on the skin, oil, and glands, and then we talked about beta-lactamase, which is the enzyme that breaks down penicillin or any other antimicrobial drugs that have beta-lactam in them. So other ways that these bacteria can, other ways that the pathogenicity of these bacteria is contributed to are toxins. It produces various different types of toxins. Staphylococcus aureus possesses lots of different toxins that will actually contribute to this pathogenicity. And they include things like cytolytic toxins, Cytolytic toxins will disrupt the cytoplasmic membranes of lots of different cells, and leukocidins can actually burst leukocytes specifically. So they can take those white blood cells and it can make them burst so that they won't be effective against Staphylococcus. We have exfoliative toxins. There are two different proteins that cause what we call the dilution of the desmosomes, or those kind of flexible junctions between the cells, and this will cause the patient's skins to separate from each other and actually to slough off. So we'll look at how the exfoliative toxin and the role that it plays in staphylococcal scalded skin syndrome. So for a lot of these toxins on here, we will actually be able to see how it directly affects the body. Toxic shock syndrome toxin causes toxic shock syndrome, which we'll talk about in a little bit. And then we have enterotoxins that they're going to mimic, and there's quite a few of them, there are about 4 or 5 of them. They're going to mimic some of the symptoms that are associated with food poisoning, because what they do is they stimulate the internal muscles to contract, and as they do that, it will cause nausea and intense vomiting, which is the same type of response or symptoms that we see with the staphylococcal food poisoning. Enterotoxins are pretty heat stable, and they can remain active at 100 degrees Celsius for up to 30 minutes. So even though we may have killed the bacteria, we will still have this enterotoxin that's left, and it can still make you sick, even with the boiling of it or the heating it up to really high temperatures. So here is a nice little chart here that's comparing the virulence factors of Staphylococcus species with whether or not it has protein A, which can be used as a structural defense against phagocytosis, coagulases, slime layers, any of these other enzymes that would help to prevent it from being dealt with or killed by the immune system, and whether or not Staph aureus has it or Staph epidermidis has it. So this is a nice little chart to kind of at a glance see who's got what. And from the looks of this chart, you can totally see that Staph aureus has many, many more of these elements of virulence or virulence factors than Staph epidermidis, which is the reason why Staph aureus is much more, as we said in the very first slide, more virulent than Staph epidermidis. So epidemiology on here. Epidemiology is basically how do we keep it from causing disease and what ways that it does cause the disease. So just kind of understanding how to track the microorganisms. Staph epidermidis is ubiquitous on human skin. It's just going to always be on there. Staph aureus is found only on moist skin folds that we typically find it. Both of the species can grow in your upper respiratory, gastrointestinal, or urogenital tracts. It's transmitted by direct contact, so just skin-to-skin touching, or mucous membrane to mucous membrane touching, or even on fomites. And what a fomite is just something that has it on it. So bed sheets, clothing, medical instruments, those will be considered fomites. Proper hand washing and aseptic techniques, which means flaming your loop, foaming in and foaming out of a classroom, or not a classroom, or the hospital room, washing your hands properly. Those are great ways, excellent ways, in fact, to prevent infection. So when we look down here, we're going to talk about staphylococcal diseases specifically right now. We're going to talk about the symptoms and prevention of staphylococcal food poisoning, and we're going to talk about different elements of it. So first type we'll talk about is the non-invasive disease, because not all staphylococcus diseases become systemic, and we'll look at those in just a little bit. So the non-invasive disease can cause things like food poisoning, and that's not as a result of the bacteria per se itself, even though you may have killed the bacteria. Remember, that's that enterotoxin that can withstand really high temperatures for a good amount of time. So it's due to that ingestion of that enterotoxin. Cutaneous diseases on here. Cutaneous diseases, Staphylococcus aureus causes what we call localized pyogenic lesions. Staphylococcus aureus can cause impetigo, scalded skin syndrome, and folliculitis on here. So scalded skin syndrome is really just a reddening of skin that happens near the mouth, and it can spread all over the body, and it has these really large blisters that have clear fluid that don't have the bacteria or the white blood cells. These are there because there's a toxin that's released by the bacteria growing in the skin, and within about 2 layers, the outer layer of the skin is going to be affected and peels off in sheets as if it has been scalded. Small red patches that can be on the face, the limbs, that we usually just see in children who don't have an immune system that's fully developed yet, we call that impetigo. The patches kind of develop into these pus-filled vesicles that usually crust over, and they have more of a golden color to those vesicles, and they're just kind of crusty, so they look a little crusty a little bit. The pus is filled with bacteria and blood, which is different from scalded skin syndrome. Staph aureus alone causes about 75 to 80% of the impetigo cases, and only about 20% of those cases actually involve streptococci. Folliculitis is an infection of the hair follicle, where the base of the follicle has been infected by the bacteria that live on the skin, and it can go into, it becomes red, swollen, and pus. Those bacteria go into that follicle. This can happen at the base of the eyelid, and that's called a sty. A furuncle is a boil that's large and it hurts a little bit, and that's just a nodular extension of folliculitis in surrounding tissue. When we have several furuncles coagulate or come together, they cause a carbuncle, which goes deeper into the tissue, and that can actually trigger fevers and chills and other characteristics of your innate immune system to kind of kick in to try to fight the infection. Now, once we have streptococcus go from these cutaneous areas, whether it's dealing with the skin or the follicle, and if it goes even deeper, then we can say that it may go systemic. So here's an excellent picture of that scalded skin syndrome. So with that scalded skin syndrome, we see how these reddening of the skin, it starts to make these blisters, and they start to leak, and then and then the skin just kind of sloughs off of itself, and it's from that exfoliative toxin that causes that to take place. This is a picture of impetigo. We usually see it and notice how it's got like kind of golden aspect to it. So it's kind of a golden crusted area. We see them on small children, and their immune system hasn't really been developed really well, and it is contagious because with just direct contact, we can another person can be contaminated. Now, as we said before, if it goes deeper, then we said that it has gone systemic. Systemic diseases means that it's no longer just on the surface, it's now into the bloodstream. One of the most talked about systemic staphylococcal disease is toxic shock syndrome, and that's really non-streptococcal. So when strains of Staphylococcus produce a toxic shock syndrome toxin, they can grow in a wound or in a vagina that's been abraded. So for women that use tampons, we saw lots of this happening in like the 70s or 80s because they were making tampons much more absorbent. But the applicator, which was used to insert the tampon, would sometimes abrade the vagina, and it would allow for, so it's a combination of the perfect storm. You'd have a small ticks or abrasions into the vagina, so the area is somewhat open, and then you have these tampons that were being left in there for a much longer period of time. It had bacteria on them just from the handling of them with their hands, with your hands. So you have those bacteria, and there's no place for them to go except for to go into the bloodstream because the tampon has not been changed very often. So we see that with tampon users for women, but we can also see it grow in wounds, and the toxin can be absorbed into the blood via the room or an abrasion in the vagina. And what we find, the characteristics of toxic shock syndrome, the non-streptococcal, are fever, vomiting, red rash, extremely low blood pressure. That's a hallmark of anything that would be considered a shock. That's a drastic drop in blood pressure, loss of sheets of skin, and this toxic shock syndrome is fatal to about 5% of patients once their blood pressure falls. And it has to fall so low that the brain, the heart, and the other vital organs don't have enough blood supply, and that's what we call shock. That's that drastic drop in blood in blood pressure on there. We can have toxic shock happen in both males and female, but as I said before, in the 1980s we saw that it was happening a lot more in menstruating women as a result of this tampon use because they were making them really, really absorbent as a result of making them really absorbent. Women didn't change them as often as they normally would, and the abrasions from the applicators, the abrasions caused by the applicators set up this sort of perfect storm for these infections to take place. So what we're looking here is toxic shock syndrome. The fatal infections not only involve this red rash that we're seeing, but also the death of internal organs as a result of that drastic drop in blood pressure. And this is just a picture of a graph that's showing the incidence of toxic shock syndrome in women and how it went down after they took superabsorbent tampons off the market. So taking those superabsorbent tampons off the markets in the early 80s, late 70s, when we were seeing lots of these things happen with women, when they said that these tampons require labeling, all of those sorts of things, and just kind of bringing awareness to it is really more of what's happened. Women, if you've started menstruating, which I'm sure most of you have, and even if you haven't, if you have a mother or a grandmother, they've probably talked about toxic shock syndrome. In fact, I remember my mother and grandmother talking about it, and that we were not allowed to use tampons, none of my female cousins or anything, because they were afraid of this happening to us, because it can be fatal. All right, so moving on. Other streptococcal diseases can cause bacteremia, which is the presence of bacteria in the blood. After staphylococci enter the blood from the site of an infection, it can go to other organs that become infected. Furuncles, vaginal infections, infected surgical wounds, and contaminated medical devices such as catheters have all been implicated in cases of bacteremia. Nosocomial infections, they used to call them nosocomial infections, they're healthcare associated infections. They account for about half of these cases of bacteremia, for streptococcal bacteremia, and this is just where physicians may sometimes fail to identify the initial site of the infection, and it's presumed to be an innocuous break in the skin, so an innocently break of the skin. Endocarditis on here. As for Staph aureus, will actually attack the lining of the heart, including the heart valves, and they can produce a condition known as endocarditis. Endocarditis, it happens typically for patients with nonspecific, what they will see will be nonspecific flu-like symptoms, and then their condition just starts to very rapidly deteriorate as the amount of blood that comes from the heart, the amount that's coming out because you have an infection of these valves, that amount has been reduced. And about 50% of patients that develop endocarditis don't make it. Pneumonia is an inflammation. The Staphylococcus in the blood can invade the lungs and cause pneumonia. It's the inflammation of the lungs and the alveoli, the air sacs within the lungs, or even the bronchioles, the airways coming in there. Once we have that infection take place, then the airways become filled with fluid. About 10% of patients with Staphylococcal pneumonia, that infection will spread to the space between the lungs and the chest wall, where the pus will build up, and it will cause empyema. Osteomyelitis is an inflammation of the bone and the bone marrow. This will actually happen as a result of a traumatic wound, or sometimes even the blood during a bacteremia case can actually enter into the site and cause osteomyelitis, and it's just the inflammation of, like I said, the bone and that bone marrow. Characteristics of this are pain in the affected bone, high fever, and in children we see it happening in regions where the long bones that are still growing, where they have well-developed blood supplies. In adults, osteomyelitis is usually just seen, most commonly seen in the vertebrae. So diagnosis, treatment, and inspection. So since we know that these Staphylococcus bacteria, all of which we talked about have been Staphylococcus, diagnosing them is fairly easy. We'll take sputum, or we can take an isolating from the blood, the pus, or other fluids, whatever sputum it is, do a gram stain and look for the arrangement. It should be gram-positive with grape-like arrangement clusters. What we find for how to deal with them is a treatment of methicillin, or we'll use vancomycin, which we use that to treat methicillin-resistant Staph aureus or MRSA. Best prevention for it is hand washing, hand antiseptics, and it's really important for healthcare associated infections that we used to call nosocomial infections here. With most of the cases of bacteremia we find are nosocomial or healthcare associated infections. Foaming in, foaming out, making sure you use proper protocol, and washing your hands often and washing your hands very well have been shown to be very well-established preventative techniques. So now moving on to Streptococcus. So I do believe with that we will start Streptococcus today. So that will be in another part that we'll talk about Streptococcus. All right, so have a great morning, noon, or night, and I will see you all or talk to you all very soon. Hope you enjoy. Bye.